like to thank Breakthrough Prize and Berkeley for hosting this symposium. So my collaborator and friend, Frank Bennett, and I received this award for the development of a drug called nusinersen, and the trade name is Spinraza. And this drug was approved at the end of 2016. And I think it's an example of a new type of precision medicine. And so this is some information from the pamphlet that comes with the medicine, the prescribing information, and you can read that Spinraza is indicated for the treatment of spinal muscular atrophy, or SMA, in pediatric and adult patients. And then in another page, you see a complicated chemical formula. There's 18 nucleotides or building blocks. So this is a type of drug called an antisense oligonucleotide. And Frank this afternoon will have a lot more to say about that. I'll mention some application of that. And you can read that nusinersen binds to a specific sequence in the intron downstream of exon 7 of the SMN2 transcript. Now, that's a lot of molecular biology terms. And I can confidently predict that in the next decade, there will be a lot more information of this type in prescribing information for new medicines. So I want to explain a little bit what these concepts mean for the non-biologists in the audience mainly. DNA, which stores the genetic information, is tightly packaged into chromosomes. Angelica talked about this morning. And so the chromosomes are present in the nucleus of the cell. And so there's something that we refer to as the central dogma of molecular biology, which is that information flows from DNA to RNA to protein. And so genes can be in an on state or an off state. So gene expression refers to this process of turning genes on. So when a gene is turned on, the information it stores is copied into a messenger RNA. So that's a single strand of RNA as opposed to the double strand of DNA. And that process is transcription, and it occurs in the nucleus of the cell. Then this messenger RNA, or mRNA, has to be exported to the cytoplasm, where it undergoes what we call translation, which is the decoding of the information. And this is done by a cytoplasmic machine, the ribosome, with the help of these transfer RNA or tRNA molecules. So the ribosome is able to translate from the language of DNA and RNA, which are, the language is written in 4 letters, nucleotides, into the language of proteins, which is written with 20 letters, amino acids. So you can see that the ribosome and the tRNAs are decoding the information in words of 3 letters in sequence, and then here's the new protein emerging. It's difficult to point with this device. The more accurate representations of the ribosome and tRNA are seen here on the lower right. How does this happen? Apologies here. So the way this translation process occurs is through the genetic code, and so this was the Rosetta Stone of life, and it earned the Nobel Prize in 1968. But so since the language of RNA is written in 4 letters, and the words of 3 letters, there are 64 combinations, and these are more than enough to code for the 20 amino acids. And there's also words that represent start and stop of translation. So going back to gene expression, in 1977, Roberts and Sharp and their colleagues discovered that genes are interrupted. So there are segments of the genes that code for proteins, and these are separated by non-coding segments. And they're called exons and introns, respectively. So you can see that the initial copy of the gene has these segments, and I use black for the exons and gray for the introns. And the process of RNA splicing is what does the cutting and pasting, so that ultimately you can produce a functional, mature messenger RNA that can then exit the nucleus for translation. This is a recent snapshot of what a spliceosome in action looks like. So it's a very, very complex machinery, even more so than the ribosome. I think it's useful to use an analogy. And so I think we can think of our genome as a library, a beautiful library. And every book in this library is a recipe book, a cookbook for how to make a protein. So we have more than 20,000 such books. And here's one. And this is the book for how to make a protein called SMN, which is important for survival of motor neurons. So we understand the language that this book, this recipe comes in. It's the genetic code. And every chapter means a particular part of the SMN protein. And the issue is that there's these other pages that are written in a different language, if you will. And in order to make sense of the recipe, we have to skip these intron pages or rip them out of the book. Sometimes a mistake is made. In the case of spinal muscular atrophy, there's a gene with a good recipe, except that the splicing machinery forgets to read chapter 7 sometimes, and therefore the resulting protein is not functional. So I also want to tell you about alternative splicing. We used to think that one gene codes for one protein, but the majority of our genes can code for 2 or more proteins that we call isoforms. So here's 4 examples. So there are 4 genes, and the boxes denote exons, and the horizontal lines denote introns. And so so when these genes are transcribed and then undergo splicing, that can happen as shown at the diagrams at the top, joining which exons are joined, or the diagram at the bottom shows an alternate pathway. So each of these examples are genes that can produce 2 mRNA and protein isoforms. And these can have similar or completely different functions, sometimes opposite functions. And so it's actually very important that we understand in detail what the types of isoforms that are made and what they can actually do. So going back to the book analogy, I'm reminded of this novel in 1963 by Julio Cortázar, an Argentine writer. And so this book has 155 chapters. There are genes that have even more chapters than that, believe it or not. And so he tells us that the last 99 chapters are expendable, and you can either read from 1 to 56 or by hopscotching through all 155 chapters. So this novel is called Hopscotch. I highly recommend it. And so this is a shot of a genome browser for one particular gene, P53, which Angelica mentioned this morning. This gene is critical to protect us against cancer. And every line shown here shows an mRNA, the structure of mRNA isoforms produced by this gene. So you can see that there are many, and that some begin or end in different places, and that some of the chapters, some of the exons are present in some of the isoforms, but not the others. And we really know the function of only a subset of these isoforms, and I think it's very important that we know much more about it. And this is just a representative gene. So I want to briefly describe antisense oligonucleotides. I think everyone knows, and it's already been mentioned today, that we have a DNA as a double helix, and what holds the strands together are the base pairing of the building blocks, A, T, and GC, because A always pairs with T, G always pairs with C. And this structure was worked out by Watson and Crick in 1953. So this is a planar representation of that. So if you know the sequence of nucleotides or bases along one strand, you can immediately infer what the sequence of bases is along the other strand. When DNA is transcribed, the messenger RNA has the same sequence of bases as one of the 2 strands of the DNA. An antisense oligonucleotide, or ASO, is a chemically synthesized short strand, a short snippet of nucleic acid, similar to RNA or DNA, but with certain chemical modifications to make it more drug-like. However, when it's synthesized, the sequence that you choose to have in there precisely determines what it's going to target. And it's going to target a particular messenger RNA in the cell, And moreover, it'll pair with a particular segment of that messenger RNA. So it's a highly precise targeting process. And of course, it won't look planar like that. It'll form some sort of helical structure as shown here. So I want to tell you about the gene that we target with nusinersen, and that's called SMN2 for survival of motor neuron 2. It turns out that we have two very similar genes, SMN1 and SMN2, but SMA patients are missing functional copies of the SMN1 gene. So they are very strictly dependent on SMN2, and if they have more copies of that, their disease is milder. This gene can produce two versions, two isoforms of protein through alternative splicing. And so at the top is the functional isoform. This is just a diagram showing you the protein has different portions of it that are important and how each exon or chapter codes for a particular segment. But this only represents about 10% of the output of this gene, and 90% is a different isoform that lacks exon 7, as you can see here. So the right end of this protein is different. And this protein is not functional. And what Spinraza does is it tricks the splicing machinery into making more of this isoform and less of that. And that way, motor neurons can be healthier because they can manufacture more of this protein that they require. So this is the chemical structure of Spinraza. And so it's an 18 nucleotide antisense oligonucleotide, and its sequence directs it precisely to this region after exon 7 and the SMN2 transcript. And normally, there's a protein that likes to bind there, which functions as a repressor of splicing with regional effects. So when this protein is bound here, it blocks the splicing machinery from efficiently recognizing chapter 7. And however, in the presence of nusinersen, this protein is no longer able to bind, and the spliceosome can now see exon 7 more efficiently. Therefore, we get more mRNA and more protein. This has to happen in motor neurons, and motor neurons are very large cells that power our muscles, and the cell body of the motor neuron is located within the spinal cord. This shows a section of the spinal cord. And what we have to reach is the nucleus of the motor neuron because that's where splicing occurs. And the way we're able to achieve that is by injecting Spinraza into the fluid surrounding the spinal cord. Patients receive 12 milligram doses infrequently, so every 4 months, initially more frequently to build up the drug concentration. I just want to discuss anticipated developments in the next decade. I think it's going to be extremely important, and this is a collective effort, to achieve a genome-wide annotation of ideally all mRNA and protein isoforms. That means understanding the structure, what parts of exons or whole exons are present or absent. That's somewhat easier, but determining the functions of those isoforms, where and when they are made, and also the relative amounts of each. And typically, you have more than 2 isoforms per gene. And so if we know that, since we have tools to be able to change the relative abundances of isoforms, if we knew what they all do, we can achieve desired outcomes for therapy or for other purposes. I think at the same time, we're going to improve our ability to understand and predict how particular mutations affect splicing. And so I expect that there will be new targeted antisense as well as small molecule therapeutics that are able to modulate the splicing process for individual transcripts. And so they can either restore correct splicing when it's disrupted by a mutation, or they can change alternative splicing to achieve a desired outcome. I'm certain that there will be continued improvements in making antisense drug delivery more efficient, more targeted, so that it can go to particular organs or tissues. Currently, that's achievable for hepatocytes in the liver, but it'll be useful to do it for many other target tissues. I think it'll become practical to do a prenatal treatment with antisense oligonucleotides by injection into amniotic fluid, like an amniocentesis, but in reverse. I think this is achievable, and that could be important for developmental disorders where things that go wrong are happening before birth. There are already some small molecules that can modulate splicing, but I think we need to know a lot more into their mechanism of action and how to make them really specific, perhaps using structural principles from studies of the spliceosome to design them more intelligently. And also there will be interesting applications of more general inhibitors as opposed to modulators of splicing. And here's 4 examples of small molecules that are undergoing clinical development. So in terms of what diseases are targetable in this way, I told you about spinal muscular atrophy, for which we already have 1 approved drug. There's also 2 small molecules that are in clinical trials with the same target. In the case of Duchenne muscular dystrophy, an antisense oligonucleotide received conditional approval also in 2016 to alter splicing, in that case normal splicing, in order to bypass another problem similar to what Jennifer was telling us about. And so that actually turns a severe disease, Duchenne's, into a milder one, Becker's muscular dystrophy. So these are other diseases for which targeted splicing modulators, antisense, or small molecules are under development at present. And so you can read about the specific instances, but at the bottom I listed many others. I think cancer, although it's a very difficult problem, also offers potential, and one idea is to change alternative splicing of a relevant gene in a way that will harm tumor cells but not normal cells. And another idea is that based on the observation that some tumor cells are more susceptible to partial inhibition of the general splicing process, and these are 2 small molecules that have been studied in clinical trials. And with that, I just want to acknowledge many people, all present and past members of my laboratory, but in particular, I listed some who worked on the development of nusinersen and or the work that preceded that, which was mechanistic work about splicing of SMN2. And especially I want to thank Yimin Hua, who did a lot of the work, and he's currently a professor at Soochow University in China. So this was at Cold Spring Harbor Laboratory. We worked very closely with Ionis Pharmaceuticals, Frank Bennett and his colleagues there. The clinical trials were sponsored by Ionis and Biogen, and also I have to thank clinical teams that conducted the trials, and more than anybody, the patients who participated in the clinical trials and their families, and also the RNA and SMA scientific communities, and finally, foundations and agencies that supported our basic work and translational work on spinal muscular atrophy. Thank you.
Yes, thank you for your phenomenal work. Would these diseases be amenable in sometime in the future to the CRISPR-Cas9 system that we heard about earlier, or are the patients going to have to get injections for the rest of their lives with these modulating molecules?
Right, thank you. I think there's different ways to approach these diseases. CRISPR-Cas9 is one way. I think what needs improvements there is efficiency. So in diseases of this type, you may want to reach close to 100% of the affected cells or just the majority, and changes have to occur uniformly. Typically, this technique is very widely used, but in the laboratory, we can select the cells that have undergone exactly the change we want. But if that cell is a motor neuron in the spinal cord, first you have to know how to do that very efficiently. And I think it will be possible. It still needs more development. But in this case, and with antisense oligos, we're targeting the RNA, so we're not making changes in the gene. And therefore, the patients continue to need the drug to maintain high levels of the protein.
Is there any work being done to make it so it will be less frequent than 4 months? Any improvements to it in the pipeline?
Right. Yeah, so the chemistry of the oligos is such that they have a very long half-life in the central nervous system. So I think it is possible that you can go to higher doses and therefore reduce the frequency with which they're given. I think that, so there was since there was no other treatment available, there was some urgency to kind of get a protocol that works and get it approved. And if you want to make changes, that will perhaps require more studies or more clinical experience. But I think it's likely to happen. Yeah. Thank you.
Thank you.